Hi everyone, welcome to today's class, Evolutionary Genomics, Building a Phylogenetic Tree. So let me walk you through the learning objectives. At first, it's very important to gain some quick background knowledge about DNA and how genetic variation works. Then you are going to learn about evolution and the construction of phylogenetic trees. The last step will be our hands-on bioinformatics experience with NCBI, BLAST and MEGA to apply the learned concepts. We will analyze the genetic data of Felis catus, which is the scientific name for the cat. Alright, let's start with the DNA. DNA has all the information that determines our traits, like the color of our eyes or how tall we are. DNA is a double helix, like a twisted ladder. It has two complementary strands running in opposite directions from each other. The building blocks of DNA are the nucleotides, and each one consists of a 5-carbon sugar, a phosphate molecule, and a nitrogenous base. We have 4 types of bases, as you may know: adenine, thymine, cytosine, and guanine, or else A, T, C, and G. A always bonds with T, and C always bonds with G. Think of the bases as the binary code of computers. They both have instruction manuals, but they use different symbols to communicate. What I would also like to stress out is that we have a lot of genetic variation and mutations. Changing a base in the DNA sequence can alter the instructions, affecting how an organism looks, behaves, and reacts. Since DNA has all the hereditary information, it's possible that we can understand the evolution of organisms. Evolution is the change in the genes of a population on Earth over time. This can lead to the formation of an entirely new species. But how do scientists figure out how certain species evolved into other species? Well, they have many ways through which they collect evidence: the fossil record, comparative anatomy, comparative biochemistry, comparative embryology, molecular biology, and biogeography. And what do scientists do with all that information? They may want to build a phylogenetic tree. Phylogenetic trees are branching diagrams that represent the evolutionary relationships among various species. These trees illustrate hypotheses about the evolutionary history. They have branches that represent evolutionary lineages, nodes where branches split, representing common ancestors, a root which is the common ancestor of all entities in the tree, and they also have leaves, meaning the endpoints of branches, representing current species. We can construct phylogenetic trees by comparing DNA sequences from different species. Our lab will be based on this, so the first thing we will do is obtain DNA sequences from species of interest, then align them to identify regions of similarity and difference. This process involves comparing the sequences base by base. And the more similar the sequences are, the more closely related the species are likely to be. What you need to do is type NCBI, which stands for National Center for Biotechnology Information. Click on it and choose the gene one right here. For this tutorial, I have chosen the NDN gene from Felis catus. Again, Felis catus is the scientific name for the cat, and NDN is just a gene name. All genes have such strange names, so don't worry about it. So here you can see some general information about the gene. Don't feel the need to understand it. You don't need to. All you need is the reference sequence of it. So click on the first one. And again, feel free to skip this information. You don't need it. You can read it if you want to gain a better understanding of the topic, but all you need to do is copy-paste this sequence. So open BLAST. Click on nucleotide BLAST, because again, we are working with genes for this tutorial. So click on it, paste the sequence, and don't change anything else because they are by default. You can maybe add a title to remember your work, but other than that, click on Blast. This may take some time because it basically goes to the NCBI website and looks for information there. But here we are. You can see here the different organisms that have very high similarity to Felis catus in terms of this specific gene. And you can also click on distance tree of results right here. And here is your first phylogenetic tree. You can see it. You can maybe change some functions to see what happens. But congratulations, you just created one. And you can go back to Blast and click on alignments and see that what it basically does is it compares the bases one by one. So I want you to do the same thing for another gene and of course play with these websites and these tools to gain a better understanding. This time I have chosen the ASIP gene for Felis catus. Again, skip this information, you don't need it. Click on the first one right here. When you find the reference sequences, go to mRNA and proteins and click on the first one because it's the standard that we use and it's the most popular one. So click on it. Again, copy the sequence and open Blast again. Click on nucleotide Blast. Again, we are working with genes. Paste your sequence and click on Blast. So again, this may take some time. And click on distance tree of results. So what I want you to see here is that this tree that we have created with a different gene is different than this tree. We can't create a phylogenetic tree based on only one gene. We need more and we need to compute this information. But it's a good practice and it's a very interesting one, I believe, to see your knowledge in action. What you can also do by using the Blast platform is use a bioinformatic tool called Mega. You can download it very easily. It is a free tool. I have already downloaded. So what we're going to do is basically create a phylogenetic tree for 5 organisms. We don't need all of them to make it a more simple tree. So click on the first 5. I am working with the NDN gene, the first one. So we have the Felis catus one, which of course has 100% similarity because it's the same gene. Sorry, it's right here, the similarity. And we are working with 4 other organisms right here, and all you need to do is download it as FASTA, as a FASTA file, aligned sequences, click on it, and here you can see the 5 sequences for our 5 organisms. What you need to do is open the Mega software, click Align, edit Build Alignment, create a new alignment, and click OK. Are you building a DNA or protein sequence alignment? We are building a DNA one, so click DNA. Now we want to insert the sequences. So we are going to click Edit and then Insert Sequence from File. So we're basically inserting the file we have downloaded as a FASTA file from Blast. So here are the sequences, you can see all of their bases. We basically don't need sequence 6 because as you can see it has like no bases at all. It is there by default, so basically we want to delete it, so click X right here and delete it. So now we want to click Edit to select all of our sequences, so we select all. And now we want to align them. Click Alignment and Align by Crystal W. We don't click the second option because it is for proteins. You can see that it has codons there, so don't confuse these two options. Click the first one. Don't change these are by default. Click OK. So now you can see that all of our sequences are aligned. And now we want to save this and export it. So click Data and then Export Alignment and click Mega Format. So we're going to save it as a demo and I'm gonna save it at Downloads. You're gonna choose the location and click Save. Protein coding nucleotide sequence data, the answer is no. We are working with DNA. So now just delete this or go back to your home screen. And now click Phylogeny right here. Click Neighbor Joining Tree because that's a very popular way of creating phylogenetic trees. Click this right here, the one that we just created. These again are by default, please don't change them, and click OK. So here you can see our phylogenetic tree and branches and the nodes and the common ancestors. You can see them all, and congratulations, you just build your own phylogenetic trees with 3 different bioinformatics tools. I hope this lesson was valuable, so stay tuned for more lessons.